Hello everyone, welcome back. Today I am here with this presentation on Tobacco Mosaic Virus. In this presentation I will discuss about the general structure of this Tobacco Mosaic Virus, its symptoms, various modes of transmission of this virus, and finally some disease control measures. In addition to this, I will try to cover the economic importance of viruses and some terms like prions and viroids. So let's dive in. So firstly, this tobacco mosaic virus. Why to study this virus? Because this virus was first extensively studied virus और इसको बहुत डिटेल में स्टडी किया गया है and this virus affects the tobacco crop which causes loss of billions of dollars, जिससे billion of dollars का loss होता है. So that's why it is very important to study this virus, and it was the first virus to be studied. So it was extensively studied virus then first to be described. और ये जो virus है it affects more than 200 species of various plants. और जो mainly हम इसको study करते हैं, it causes a disease that is called tobacco mosaic disease, and it can affects more than 200 individual species. 200 से ज़्यादा individual species को भी affect करता है angiosperms की. If we talk about its historical background, I have already discussed in previous presentation. So there were various scientists who contributed in this field. First was Adolf Meyer, who reported this disease. He was the first person to report this disease, and this disease can be transferred between the plants. So he observed this thing, that this plant is suffering with this disease, जिसे हम कहते हैं tobacco mosaic disease. Then second was this Bijering in 1889. He used this bacteria-free culture by using bacterial filter, and he observed that this filtrate was capable of causing infection in the healthy plant. So he gave a term that is contagium vivum fluidum, means contagious vital or viable fluid that can cause infection. Then the first evidence of existence of virus particle, means the discovery of virus, was done by D. Ivanovsky, was a Russian scientist in 1892. W. Stanley, he was the first person to isolate and crystallize this virus, and these viruses are active or remains active after crystallization के बाद भी active रहते हैं. And finally there were two scientists named as Conrad and William. They reported or observed that जो भी TMV virus है, इसके जो parts हैं, like we have capsid and RNA, तो ये separately बनते हैं, and after that they gets assembled into a functional unit. So जो separate units are they assembled and they form a functional unit. So this was observed by these two persons named as Conrad and Williams. If we talk about its virus structure, so it is basically a rod-shaped structure. In symmetry I have discussed that this virus shows helical symmetry, and it form a rod-like structure जिसमें central core होता है that contains nucleic acid as RNA, जिसमें RNA present होता है genetic material, and that is covered by the proteinaceous covering जिसे बोलते हैं हम कैप्सिड, और जो individual units हैं कैप्सिड की उन्हें कहते हैं कैप्सोमियर्स. If we talk about its length, so इसकी जो length होती है that is near about 300 nanometers, and its diameter is near about 18 nanometer, means it is 18 nanometer wide. If we measure it in Armstrong, so it is 3000 Armstrong, or we can say 300 nanometer, and its length, nanometer in length, and 180 Armstrong, that we can see here, this 180 Armstrong or 18 nanometer in diameter. So it contains this central helical single stranded RNA, that is nucleus RNA, and it is covered from outside by the proteinaceous covering जिसे बोलते हैं हम कैप्सिड और प्रोटीन कोट. इट इज मेड अप ऑफ दीज सब यूनिट्स जिन्हें कहते हैं कैप्सोमियर्स, और इसका जो नंबर है दैट इज डेफिनेट इन TMV, that is near about 2134, और in some books it is written as 2130. So इतने इसमें कैप्सोमियर्स होते हैं. So this tobacco mosaic virus is a rod-shaped virus which contains RNA. इसमें RNA होता है, if we talk about length, so it is 300 nanometer, or in Armstrong it is 3000 Armstrong, and it is 18 nanometer wide, or we can say 180 Armstrong wide in diameter, and its molecular weight is approximately 39 million daltons. ये इसका molecular weight है. It consists of outer covering जिसे हम बोलते हैं capsid and inner single stranded RNA molecule. So nucleoprotein particle है. और जो capsid है that makes maximum part of this virus. It contributes about 95% of the virus weight and contains near about 2134 protein molecules or capsomeres, which are helically arranged in a staircase manner. Then this each capsomere has 158 amino acids. जो ये capsomere है इसमें 158 amino acids होते हैं और इसका जो weight होता है that is approximately 17,500 daltons. If we talk about RNA, so its contribution is very less. That is, it forms about 5% of the virus weight. Virus का weight का जो contribution है 5% weight बनाता है, and it has around 6,500 nucleotide. और इसमें जो न्यूक्लियोटाइड्स हैं वो 6,500 होते हैं और ये अरेंज होता है हेलिकली. इफ़ वी टॉक अबाउट इट्स diameter in the central region, so it forms about 4 nanometer central cavity, or in Armstrong we can say 40 Armstrong cavity, the central cavity. In this cavity this RNA is present. In this diagram we can see here, so central cavity of 40 Armstrong, or we can say 4 nanometer. In this cavity this RNA is coiled around. So RNA is protected by this capsid from cellular enzymes. जब ये host के अंदर enter करता है तो जो RNA है that is protected by capsid from the cellular enzymes. Next comes the symptom of tobacco mosaic disease. So it shows various symptoms on the plant after its infection. So most common symptom is it gives patterns of white and green dark patches that gives the appearance of a mosaic pattern. इसीलिए इस disease को कहते हैं mosaic disease of tobacco. So जो white blisters और dark green patches इसमें दिखते हैं that we can see in these images. So ये जो blisters दिखते हैं इसमें white patches और dark green patches, and in this we can see here जो ये vein है young plants में it becomes decolorized. So और ये जो patterns इसमें दिख रहे हैं whitish और greenish patterns, so these forms a mosaic pattern. So these are some common symptoms. If the plant is suffering with severe infection, तो जो plant की growth है वो रुक जाती है, plants become stunted. So firstly, जो ये वायरस है ये मोस्टली अफेक्ट करता है मेंबर्स ऑफ फैमिली सोलेनेसी. सोलेनेसी में काफ़ी सारी वेजिटेबल्स आती हैं और वाई वी कॉल इट एज सोलेनेसी बिकॉज इन द मेंबर ऑफ दिस फैमिली जो प्लांट्स है दे कंटेन स्वोलन ओवरी. इफ यू टॉक अबाउट अर्ली सिम्टम्स, इनिशियल जो सिम्टम होते हैं, सो लाइट डिस्कलरेशन अलोंग द वेन्स. जो वेन्स होती हैं वहाँ पे जो ग्रीन कलर है वो डिस्कलर हो जाता है in the young leaves. So this is the first symptom. Then mosaic pattern that I have already discussed. जो leaves हैं उनके ऊपर light और dark green color के patterns बनते हैं, patches बनते हैं with distortion and blistering, जिससे जो leaves है वो deformed हो जाता है. So that's why this is called tobacco mosaic disease. If we talk about its growth after infection, अगर अर्ली स्टेजेस में इन्फेक्शन हो जाता है प्लांट में तो जो प्लांट्स हैं वो ज़्यादा ग्रो नहीं कर पाते हैं, दे रिमेन स्टंटेड, मीन्स dwarf in their size, and later stages infection show minimum effect. अगर initial stages से plant survive करता है, healthy रहता है, later stages में ज़्यादा इसके symptoms नहीं दिखते हैं plant में. In case of severe infection, जब बहुत ज़्यादा infection रहता है, plant become puckered. Puckered means having deformity. जो leaves हैं इसके ऊपर छोटे छोटे ऐसे deformed structure बनते हैं, that is called puckered structure, and narrow, and the whole plant may become malformed. तो जो plant है उसकी growth stop हो जाती है जिससे जो leaves हैं वो deformed हो जाते हैं, that causes loss of yield. If we talk about temperature effect, तो जो temperature का बड़ा important role इसमें रहता है. जो ये legends हैं, मेन जो ये patches हैं ये restricted रहते हैं plant में, एक ही जगह तक रहते हैं, localized रहते हैं अगर temperature 28 डिग्री सेल्सियस से कम होता है. If this temperature exceed 28 डिग्री सेल्सियस, so ये जो patches हैं ये पूरे के पूरे plant और leaves में spread करते हैं, so it becomes systemic. Systemic means spreading. So above this temperature 28 डिग्री सेल्सियस ये जो blisters हैं, patches हैं वो spread करते हैं. So these are the symptoms of this disease. Next comes transmission. So there are various modes of transmission of this virus. First is sap transmission. अगर जो प्लांट किसी जगह से इंजर्ड है और जो डिजीज प्लांट है उसका जो सैप है, सेल सैप दैट कम्स इन कॉन्टैक्ट विद दिस प्लांट, हेल्दी प्लांट्स, इंजर्ड पार्ट, सो ये जो प्लांट है इट गेट्स वायरल इन्फेक्शन. The next is mechanical transmission. So we use various tools in the field of tobacco. तो ये जो tools हम use करते हैं, if it is touched with the diseased plant, अगर ये जो plant infected है virus के साथ उसके contact में आता है, and after that we will touch this healthy plant with the help of this tool, so the virus gets attached to that plant that can cause infection in healthy plant. So by rubbing infected sap on healthy plant, and this can also spread through agricultural tools. जो हम कृषि में टूल यूज़ करते हैं, औजार यूज़ करते हैं, उससे भी ये ट्रांसफ़र होता है. नेक्स्ट वेक्टर्स. सो मोस्टली इसमें कोई ट्रू वेक्टर्स नहीं होते, बट व्हेन द plant sap is sucked by insects, so these insects can also act as vectors. So ये insects एक diseased plant से healthy plants पे जाएंगे और infection spread कर सकते हैं. Next comes dodder. Dodder is a parasitic plant that is commonly known in the scientific name is Cuscuta species. So this also affects the plant, and it can also cause this spread of this virus. It also directly affects the plant growth. This is a parasitic plant. It obtains nutrition from this plant. So this is called dodder plant, or you can say Cuscuta. It is a parasitic plant. It is also responsible for spread of this tobacco mosaic virus. Next comes seeds. Seeds अगर healthy हैं तो वो disease को spread नहीं करती, but if seeds are means if seed coat is damaged or it contains some sort of wounds, so that through that wounds virus can enter inside the cell, and that can cause disease in the growing plant. So these are various modes of transmission of this virus, tobacco mosaic virus. So they are seeds. So if these seeds are healthy, then they are not spreading this infection. But if these seed coat are damaged, if the seed coats are damaged, then there are chances that virus can enter inside the seed and can cause infection to the growing plant. Next comes disease control. How we can control the spread of this disease. So firstly, we have to identify the diseased plant, and we have to uproot them and discard them properly. तो फर्स्ट मेथड यही है कि जो डिजीज़ प्लांट है उसको हम आइसोलेट कर दें हेल्दी प्लांट से और उसको कम्प्लीटली डिस्कार्ड कर दें. वी हैव टू मेंटेन द सैनिटेशन ऑफ फील्ड्स को क्लियर करना पड़ेगा. जो भी डिजीज़ डेब्रीज है उसको आइसोलेट करना पड़ेगा. देन वी हैव टू सैनिटाइज आवर टूल्स. वी हैव टू मेंटेन द हाइजीन, एंड वी कैन यूज़ रेजिस्टेंट वैराइटीज़ ऑफ टोबैको जो डिजीज़ के अगेंस्ट रेजिस्टेंट शो करती हैं. So first step is roughing. Roughing means removal and burning of diseased plant. तो जो diseased plant है, infected plants हैं, उनको हमने separate करना है, isolate करना है, उनको हमने dispose of करना है properly. Next comes field sanitation. So maintain cleanliness in the field. That is very important. We have to maintain hygiene, means we have to clean our hands and our tools after handling the infected plants. Then we can use certain disease resistant varieties जिस पे इस डिज़ीज़ का कोई ज़्यादा फ़र्क नहीं पड़ता है. And we can use some genetic resistant methods, so that is used by some professionals. So use coat proteins or replicates to develop plant resistance against this disease. So these are some methods by which we can control the spread of this disease. Next comes economic importance of viruses. So this importance covers both aspects of viruses, that is useful aspects as well as harmful aspects of viruses. Mostly viruses are known to cause various human disease and disease in plants, but they are also utilized or exploited in the field of molecular biology. So we'll discuss these points one by one. So viruses are known to cause various diseases, and they also plays very important role in understanding biological phenomena or in research related to biology. Firstly, we'll discuss about the useful applications of viruses. So study of molecular biology, that is DNA replication, RNA processing, protein transport, specificity. For these to study these complicated processes, we can use viruses because viruses contain only protein and DNA, so it is easy to deal with. Next, जो concept है split genes का, means genes, as we know that genes contain coding segments as well as non-coding segments. तो ये जो concept है, that is we have a gene which contains introns and exons, so that is called split gene. introns and exons. Introns are non-coding sequences, means intron word is related to intron trash, that is not useful. Then exon, they are coding sequences. So this concept of split gene was also studied in viruses, that is adenovirus. Then next we have various viruses like bacteriophages that are used as useful vectors or carriers of DNA in case of biotechnology. And they are also utilized for the formation of various vaccines, to develop various vaccines against viral diseases. Then nowadays this is trending field, that is virotherapy, gene therapy or phage therapy. These are some useful aspects of viruses. So this is the concept of split gene. So it contains coding as well as non-coding sequences. Exons are the coding sequences, introns are non-coding sequences, and the removal of these introns to form a complete functional unit. So this is called RNA processing. इस process को कहा था RNA processing. We can say RNA splicing also. That forms a complete mature mRNA that will finally form a protein. In case of this viral vaccines, or we can say virotherapy, so these viruses are, their properties are utilized to treat various diseases, like we have cancer and other diseases. So these diseases can be treated with the help of viruses. So these viruses infect the tumor cells, and this infection spreads, and these infections spread and tumor cells are killed by viruses. So after killing the viruses, these tumor cells, the antigens are released and systemic anti-tumor immunity is amplified. तो जिसकी वजह से जो immunity है against these tumor cells that is obtained. Some harmful activities, जो viruses हैं वो बहुत सारी बीमारियां कॉज करते हैं humans में. बहुत सारे epidemics कॉज हुए हैं viruses, like we have smallpox, then COVID-19 जो recently एक बहुत ही बड़ा एक epidemic था, which causes death of millions of people throughout the world. Then AIDS claimed more than 34 million lives till date. ये data ज़्यादा है, this is somewhat older data. Then we have SARS, that is a group of viruses that causes severe acute respiratory syndrome. So it was also causes reason of various respiratory disorders in Asian countries. Then Ebola disease, जो कि African countries को affect किया था जिसने. Then we have various animal diseases such as rabies, AIDS, chickenpox, smallpox, dengue, hepatitis, measles, mumps, polio, SARS, smallpox, warts, etc. So these all diseases are caused by viruses. In case of plants, it causes mosaic diseases and stunting of growth, ring spotting, top necrosis, yellowing, vein clearing and blossom breaks, means जो flowers हैं वो जल्दी ही गिर जाते हैं. So this is a table which shows the various diseases in plants. Like we have cauliflower mosaic disease that is caused by cauliflower mosaic virus, then cucumber mosaic disease, leaf curl of tomato, mosaic disease of sugarcane, then potato mosaic disease in potato that is caused by potato virus XY, then rice tungro disease that is caused by rice tungro virus. और इसका जो transmission है that it is through these aphids and grasshoppers. Then we have tobacco mosaic disease that I have already discussed. Then tomato spotted wilt. So tomato spotted wilt virus, इसमें जो plant होते हैं tomato plant, they gets wilted due to this infection. Then comes disease caused by viruses in humans. So it causes viruses cause AIDS, then chickenpox, dengue, genital herpes, hepatitis, herpes simplex, then measles, mumps, poliomyelitis, rabies, smallpox, viral gastroenteritis, and warts and yellow fever. So these are diseases and various parts are affected by these viruses. And transmission, there are various modes of transmission. So there are some common diseases which are caused by viruses in humans. Next comes viroids. So these are again infectious single-stranded circular RNA particles. They contain only RNA and they are known to cause various disease. So these are known as viroids. So they are simple RNA pieces which are single-stranded circular RNA pieces, and they were first observed by discovered by T.O. Diner in 1971, and it was associated with one disease that is called potato spindle disease. So जो potatoes होते हैं, they become spindle in shape and they are infected by this viroids. Viroids are simply circular single-stranded RNA molecules They are circular single-stranded RNA molecules of low molecular weight known to cause disease in plants, and these are known as viroids. They are smallest known pathogens. They do not code for any proteins, and they multiply by one method that is called rolling circle mechanism of RNA synthesis. The diseases they causes, what the common one is the kadang disease of coconut. So here the coconut size becomes reduced because of this disease, and it also affects the plant growth and plant's morphology. Leaves gaps wilted or dried up. They show necrotic spots, and this is called kadang disease of coconut. It is caused by viroid. Then there is second disease that is called chrysanthemum stunt disease. Here this flowering plant becomes stunted. It is unable to show full growth. So this is again a disease caused by viroids. Next comes prions. These are simply infectious protein particles. इसमें सिर्फ प्रोटीन के जो particles they are known to cause various diseases. These are known as prions, and they were first observed by Prusiner in 1982. So they were discovered by Prusiner in scrapie. That is a disease of sheep, which is neurological disease of sheep. So infectious particles that are entirely made up of protein. So we can simply say जो prions हैं वो प्रोटीन के छोटे particles होते हैं जो disease cause करते हैं. They do not contain any nucleic acid. इनके पास कोई भी nucleic acid नहीं होता है. They are highly heat resistant, affects nervous tissue, and results in various diseases like we have bovine spongiform encephalitis, that is abbreviated as BSE. It is commonly known as mad cow disease. So bovine spongiform encephalitis ये affect करता है cows के brain को, जिससे एक disease होता है जिसे कहते हैं हम mad cow disease. Then we have scrapie disease of sheep. जो sheeps होती हैं उनमें scrapie disease होता है. That again affects the skin and its nervous system. Then there is one disease that is called Kuru, and then Creutzfeldt-Jakob disease, which is abbreviated as CJD syndrome in humans. That again affects our nervous system. The next comes one topic that is related to the purificatory power of Ganga Jal. As we know that Ganga Jal is very important according to Hindu mythology. It is a religious water and is considered as sacred in Hinduism, symbolizes spiritual purity and cleansing. It is used in various rituals and rites. जो भी हम कुछ rituals करते हैं उसमें हम इसे use करते हैं. ऐसा मानते हैं कि जो गंगा जल है वो हमारे sins को clear करता है. It wash away the sins. Scientifically, if we talk about it, so there are two renowned institutes of India. One is NERI, that is NWRI, National Environmental Engineering Research Institute, and second is IIT Kanpur. They observed that this water contain high concentration of viruses which are known as bacteriophage. So this is one of the scientific reason for its purity. तो जो भी viruses हैं ये bacteriophages हैं, ये bacteria और जो contaminants है इसमें grow नहीं करने देता है. So this is one of the reason that जो गंगा जल का पानी है वो pure रहता है. So National Institute of Engineering Research Institute. Then we have this this is present in Nagpur, and IIT Kanpur. इन्होंने report किया है जो water है गंगा का that carries high concentration of bacteriophage, which is responsible for purification power of this गंगा जल. And it is harmless. जो ये भी bacteriophages हैं ये harmless होते हैं humans के लिए. ये viruses human में कोई भी disease cause नहीं करते. So this is one of the reason that makes the power that makes the Ganga Jal to remain pure for longer duration. So may account for the purifying properties of Ganga Jal. So this was all about for our discussion about this tobacco mosaic virus, its structure, its symptoms, then transmission and disease control. Then we have discussed about economic importance of viruses and prions, viroids. I hope you will get some idea from this presentation. If you have any questions, queries, and any sessions, give it in comment section. Thanks for watching. Have a great time.